I'm Thomas Wollmann. I'm medical computer scientist, co-founder of PW Solutions and PhD student at the Biomedical Computer Vision Group in Heidelberg. In my company and in my PhD, I'm heavily working on machine learning, especially on deep learning. My whole PhD is dedicated to deep learning, especially detection and segmentation in microscopy images. Therefore, I have now about 3 years of experience in deep learning, and I'm happy to help the iGEM team Heidelberg with their project. To my point of view, deep learning is a very strong function approximator which can be used at a variety of problems. It is very good at feature engineering and pattern recognition, but a very bad classifier. However, if we have good features, we don't need a good classifier, and it's a multi-purpose tool for biology and other fields. And a main problem of deep learning is that it's just successful in supervised learning. There are still major problems in generative modeling and in reinforcement learning and in weak supervised learning, which is still an active area of research, but nevertheless interesting. Another problem of deep learning is that it's very label intensive, and also it requires a high computational cost. However, nowadays we get stronger compute in all the different data centers in universities and especially in companies. Therefore, we can expect that in future we can reduce this problem and use this technique for almost every function approximation task. Pattern recognition is a frequent task in biology, and especially segmentation, classification and clustering is already heavily used in experiments to verify and to generate hypotheses. And in the current information age, high content and high volume of data requires intelligent algorithms. And the trend is that in future data sets will grow even further, and with these models like deep learning and other machine learning algorithms, we can get insights in biology not by verifying observation, but also generating hypotheses and learning from the models what they learned, and improve our overall knowledge in biology. The short answer is yes. The long answer is it depends on the problem. Deep learning algorithms are very good at finding patterns in data. However, we have to find the correct distributions from where we can generate these patterns. And there are still some problems. For example, if we look at GANs or if you look at pixel CNNs, that they tend to be very unstable in practice and only work on toy datasets. But in the next weeks or months, I expect major improvements with their techniques, because it's one of the hottest topics in deep learning right now, and deep learning is kind of the hottest topic in computer science right now. And if these models work, then we can save a lot of wet lab experiments which saves time and money, and since saving time and money is always what people want, people will try to get generative models to work. Generative modeling can be done in two ways. You can manually design your generative model and then sample from it, or you can learn your model. I think to explain the complex structures in biology, we can't just rely on manual modeling and have to go to trained models, and therefore skipped the manual model design. However, current trainable models still lack stability and scalability to large data sets, and especially on large structured output, and therefore there still has to be major work done on improving these algorithms to make them useful in practice. An additional task for the community is to get insight into the models. Just training a generative model and then generating samples from it can help for generating products, but not to make major steps in science. Therefore, we need to get better insights into these generative models and learn from them what is the biology behind our models. Since we want to have a generative model, just using no generative model is very problematic. However, you can use a hybrid approach, therefore just use weak labels for training, and using a generative model to boost your classifier with weak labels. Another option is to use a genetic or evolutionary algorithm to generate samples and then train a classifier on it. However, the future goal should still be to have a full generative model, because then you can look at the model and sample infinite samples from it. Since this is very challenging because of technical and theoretical issues, it's probably a short-term solution for the task of protein design.